Ayan. Okay, so first is phosphoglycerides, that they are also known as glycerophospholipids. Now, phosphoglycerides are also known as glycerophospholipids. Ayan. Okay? Now, remember, no, na if you remove, no, one fatty acid, let's say from carbon 1 or carbon 2 of the glycerol. So, let's say ito yung fatty acid. Yan, nakita niyo yung ano. Siyempre, hindi nakikita, no? Yan. Ayan, so, ito yung fatty acid groups, no? Lakihan natin para mas makita ng mga nasa physical world. Ayan. So, yung ayan. Okay? Ayan. So, this is the glycerol moiety. Hindi pwedeng pagsabayin. So, this is the glycerol moiety. You have fatty acids attached to carbon 1 and 2 of your glycerol. Okay? And then you have the phosphate group. Tapos you have the alcohol group. No? So, para maliwanag, gawa na lang ako ng whiteboard. Ayan. Para makikita ng lahat. Para fair. Alright. So, dito tayo mag-drawing ngayon. Ayan, black, para mas kita. So, you have your alcohol. So, CH2OH. As you have here the fatty acid. Ayan. CHO. Tapos COR. Tapos you have here CH2O. Tapos phosphate yung nakakabit sa kanya. Tapos another O. Tapos this is the alcohol group. Okay? Ayan. R na lang yung ilalagay ko, no, for the alcohol group. No? So the alcohol group can be any of the following. Ayan. Ayan. It can be any of the following. You have your ethanolamine. Okay. You have your ethanolamine. You have your choline. You have your serine, inositol, and glycerol. All right. Ayan. So again, this is the structure of your glycerophospholipid or your phosphoglycerides. No? Ang pangit pala yung sulat ko. Or phosphoglycerides. Glycerides. All right. Now, if I remove a fatty acid in carbon 1 or carbon 2 of the glycerol, nakita pa dyan. Yan. So, yung carbon 1 and carbon 2 of glycerol, let's say tinanggal ko ito, or tinanggal ko ito, okay, isa lang yung tatanggalin ko, isa lang. That is what you call a lysophosphatide. Okay, so lysophosphatide or lysophosphatidic acid, okay, is a phosphatidic acid or it is a glycerophospholipid with only one fatty acid attached to glycerol 1 or 2. Okay, to carbon 1 or 2 of glycerols. All right? Ayan, ulitin natin. A lysophosphatide, no? Lyso, no? Lyso means may lysis na hydrolyze. Okay? Ayan. So, a lysophosphatide is basically a glycerophospholipid or a phosphoglyceride containing only one fatty acid either in carbon 1 or carbon 2 of the glycerol. Okay? But there is the phosphate group and the alcohol group. All right? So, that is what you call a lysophosphatide. Okay? Gets? Yan. Ngayon, if kompleto naman, no, kapag kompleto yung ano, yung components of your glycerophospholipids, that is what you call a phosphatide. Andito na lang natin sulat. Phosphatide naman siya. Phosphatide or phosphatidic acid. Alright? Ayan, so phosphatidic acid. Ayan. So, syempre, pag phosphatide, ayan yung anion form. Kapag phosphatidic acid, that's the acid form. Alright? Ayan. Now, if ethanolamine yung nasa R group, ayan, kita ba ang blue? Kita ang blue, no? Ayan. Okay. So, itong R group na ito, it can be any of the following. It can be ethanolamine, it can be choline, it can be serine, it can be inositol, it can be glycerol. Alright? Yan. So, yung magiging pangalan of your phosphatide will be this, no? Phosphatidyl, phosphatidyl, tapos the name of the alcohol. Okay, so for example, phosphatidyl, ethanolamine. Ayan, okay? Ayan, kasi phosphatidyl in ethanolamine. Tapos ito naman ay, sulat na lang muna natin to, choline, serine, inositol, and then glycerol. Ayan. So lahat yan lalagyan lang natin ng phosphatidyl. Ayan. So phosphatidyl, choline, phosphatidyl, serine, phosphatidyl, inositol, and phosphatidyl glycerol. Ayan. Clear? Clear? Ayan, di ba? Dali lang. Ayan. So all of these are phosphatidic acids. Ayan. So para malinaw, lahat ng ito ay phosphatidic acids. Alright? Clear? Ayan. So kunwari, tinanggalan ko ng isang fatty acid, yung phosphatidyl ethanolamine. Okay? It becomes lysophosphatidyl ethanolamine. Alright? Nagets na yung pattern? Ayan. Ganun lang. No? Yeah, so that is true for all of these phosphatidic acids. Alright? Gets? Okay. Now, phosphatidyl ethanolamine actually has another name. Okay? Ayan. So, yung other name ng phosphatidyl ethanolamine ay si cephalin. Ayan. So, this is your cephalin. Tapos, your phosphatidylcholine is your lecithin. Correct. Tapos, your phosphatidylserine, yan na yun. Phosphatidylinositol is inositide. Yan. And phosphatidylglycerol, yun na yun. No? Ayan. Wala naman ang kakaiba sa kanya. Alright? Ayan. So, remember the other names as well. Okay? Ayan. Now, your glycerophospholipids, no? Alam ko pwede ako mag-down dito eh. Or dito na. Okay, so your glycerophospholipids, sulat tayo ulit. Glycerophospholipids can be any of the following, no? Yeah, so your glycerophospholipids can be your phosphatidic acids. Ayan. So ano-ano yung mga phosphatidic acids natin? You have your, ayan, so gawa na tayo ng diagram, no? Ayan, you have your cephalin, you have your lecithin, you have your phosphatidyl, serine, you have your inositide, and you have your phosphatidyl glycerol. Ayan. Now, aside from phosphatidic acids, no, your glycerophospholipids can also be cardiolipin. Ayan. So, cardiolipin. Yeah. So, ano naman yung cardiolipin? Okay? So, the cardiolipin is basically, no, your phosphatidic acid, glycerol, and another phosphatidic acid. So, lagyan natin ito ng code, PA. Phosphatidic acid is PA. Okay? Si cardiolipin, that's just phosphatidic acid plus glycerol plus another phosphatidic acid. Alright? Ayan. So to see, no, the structure of your cardiolipin, ayan. So this is your cardiolipin, no? It is a diphosphatidyl glycerol lipid. Okay? Kaya nga sabi natin kanina, phosphatidic acid, PA. Ayan. So ito yung PA, ito yung glycerol, and then another PA. Ayan. Ito yung isa pang PA. Ayan. Ayan, nakita nyo, no? Na PA, dalawang phosphatidic acid on both ends of the molecule. And then they are connected by glycerol. Okay? Now, remember that cardiolipin can be found in the inner membrane of mitochondria and bacterial membranes. And another, a very important thing about cardiolipin is that it is actually an antigenic phosphatidic acid. No? This one is an antigenic phosphatidic acid. Okay? Ayan. So anong ibig sabihin natin ng antigenic phosphatidic acid? Remember, antigens are the ones attacked by antibodies, right? Ayan. So ibig sabihin, this can cause, no, an inflammatory response, no? Ayan, when certain bacteria, no, are present in our body. Okay, yung nade-detect ng katawan is actually the cardiolipin. Okay? Ayan, so that's another thing to remember with cardiolipin. Alright? So aside from cardiolipin, meron din tayo plasmalogen. Ayan. So with plasmalogen, no, sige po. Sige po. Okay po. Ayan, so the plasmalogen naman is either a phospholipid characterized by it is an ether phospholipid characterized by the presence of a vinyl ether linkage at S1 position of the glycerol. So anong ibig sabihin nun? Here you have your phosphatidic acid moiety. Ayan, di ba? Yan yung phosphatidic acid moiety. You have the glycerol, you have the fatty acid, you have the phosphate, you have the alcohol. Okay? Yan. However, instead of having an ester linkage, no, you have an ether linkage. Ayan. So, ano yung itsura ng ether linkage? ROR. Okay? Ayan. So, you have here the ether linkage. Ayan. ROR. Alright? Ayan. So, this is your plasmalogen. Now, this one is found in many tissues, mainly in the nervous, immune, and cardiovascular system. They protect mammalian cells against damaging effects of ROS. Now, anyone who is familiar with ROS? ROS is your reactive oxygen species. Okay? So pag sinabi ninyong reactive oxygen species, these are actually your hydrogen peroxide, your superoxide, O2, no? Ayan. Anything that can destroy the cell membrane. Okay, yan. Pwede siya mag-cause ng leakage of the cell membrane. So, your plasmalogen is able to neutralize that because of the presence of this bond. Meron siyang unsaturation, no? At this portion, no? It's an ether linkage as well. Now, take note of the name, no? Dalawa yung pwede natin gamitin. For example, mali ito, no? Hindi yan ethanolamine yan. So, the alcohol here is ethanolamine. Okay? So, ang tawag natin sa kanya ay phosphatidal ethanolamine. Ayan. Phosphatidal ethanolamine. So, take note of the difference, no? Pag phosphatidic acid, okay? Pag phosphatidic acid, phosphatidyl ethanolamine, okay? Pag plasmalogen, phosphatidal ethanolamine. All right? Yeah, so another term that we can use is this one, plasmenyl, no? Ayan. So plasmenyl ethanolamine. So that will indicate that you have a plasmalogen. Okay? Ayan. Hold on. May mga nag-chat pala. Okay. Okay na pala. Nasagot ko na yun. Ayan. So let me go back to our whiteboard. Andito pala yung whiteboard. Ayan. So remember this diagram para mas madali, no? Yeah, so 'di ba, you have your triglycerides, no? Iba 'yon, 'yun yung kanina, okay? You have your simple lipids, no? Tapos may mixed triglycerides din tayo. And then we are in phospholipids, no? Under phospholipid meron tayong phosphoglycerides and sphingolipids. Phosphoglycerides are also your glycerophospholipids, no? They can be phosphatidic acids, PA, no? Yung phosphatidic acids or PA na ito can be cephalene, lecithin, phosphatidylserine, inositide, or phosphatidylglycerol. Okay? Tapos, we also have your cardiolipin. Okay? So, cardiolipin is a phosphatidylglycerol. Okay? Dalawang phosphatidic acid moieties yung meron siya. And then next, we have your plasmalogen. Okay? Now, remember that plasmalogen, no, do not have the ester linkage, but instead it has the ether linkage, no, at SN1 of glycerol. Okay? Yeah, so yung SN1, it only means the carbon 1 of glycerol molecule. Okay? Now, aside from glycerophospholipids, no, meron pa tayong sphingolipids. Ayan. Sphingolipids. Ayan. So, yan naman yung other half of the branch. Okay? Your sphingolipids. Okay? Now, what are these sphingolipids? So, lipat tayo ulit dito. Ayan. So, your sphingolipids, no, act intracellular. Teka lang. Kanina pa ako na, ano dito. Kanina pa ako nababother doon. Okay, ayan. So sphingolipids act intracellularly as secondary messengers, no, in signal transduction. And they are also membrane component. And probably you are familiar with sphingolipids, no, because of a very important function in signal transduction, particularly nerve signal transduction. Ano yung naalala ninyo with regards to sphingolipids? Remember yung yung myelin sheath, 'di ba? Yung myelin sheath ay naglalaman ng sphingomyelin, okay? Now, the sphingomyelin, no, is important, no, in kunwari, ay, ano ba yun? Ayan. So, ito kunwari yung nerves natin, di ba? Meron tayong meron tayong mga myelin sheaths, di ba? Ayan, naka-cover doon sa nerves, no? And it is said na with this myelin sheath, mas faster yung transduction of signals, okay? Kasi tumatalon lang yung signals doon sa myelin sheath, okay? Kapag na-remove yung myelin sheath, no, mas mabagal yung signal transduction, okay? So that's the importance of your sphingolipids. Sila yung nandito, no, sa nerves. All right? Yeah, now your sphingolipids, no, they are characterized, no, or they are lipids containing the amino alcohol that is known as sphingosine. Okay? Yan. So ito yung structure of the sphingosine. It's quite a big molecule. Okay? So lakihan pala natin kasi hindi natin nakikita yung molecule. Ayan. It's a big molecule. So just in case, no, para hindi, kasi sa pag nagre-review kayo, minsan ang dami-dami mini-memory sa structure, di ba? So, ang technique is wag ka mag-memorize ng structure, no? Ayan, be familiar with the structure. Ang labo no? Lagi ko sinasabi sa mga students yan sinasabi ko sa kanila, do not memorize the structures, do not memorize formulas. Be familiar with the formulas. So, ayan. So, what do I mean by that? Ganito lang gawin ninyo, no? So, whatever structures you can memorize, no? So for example, kanina, ethanolamine, madali lang i-memorize, no? May ethanol, may amine, okay? lagyan pa natin ng choline. Okay? Choline, madali lang din memorize, no? Lagyan ng methyl groups yung nitrogen, yun na yun, si choline na yun, no? Si nitrogen ng ethanolamine, that's already your choline. And so on and so forth, no? Madali lang. Now, for those which you cannot memorize, do not attempt to memorize. Okay? Be familiar. Ganito. Pag nakita ko to, okay, hindi ko siya i-memorize, no? So, ibig sabihin, pag nakita mo isang bagay na hindi mo alam, malamang siya yun. No? Ayan. Ganun yung technique. Okay. Ayan. So, di ba? May nakita ka eh. Hindi ko alam to eh. Okay. Ito yung alam ko, eliminate. No? Ayan. So, yung hindi mo alam, yun na yun. Okay. Ayan. So, di ba? Dali lang, di ba? Ako rin kasi, hindi ako magaling mag-memorize. So, I will just tell you that this is the structure of sphingosine. Okay? Ayan. So, it's a large molecule. So pag nakita kayo ng malaking molecule na may nitrogen, na may OH, no? It is probably sphingosine. All right? So remember that sphingosine is an amino alcohol kasi meron siyang amino group. Ayan. Unfortunately, babalik siya sa maliit. May amino group siya, may alcohol group siya. All right? Ayan. So it's an amino alcohol. Now, when I connect, no, a fatty acid, no, ayan, maglagay ako ng fatty acid, this one, to your sphingosine, ito yung sphingosine, ayan, that is your ceramide. Okay, ayan, so, ayan memorize na to, ayan. Meron structure na hindi mo kilala, okay, most likely that is sphingosine, kabitan mo ng fatty acid, then that is ceramide, okay? Ayan, so that's an example of your sphingolipid. All right? Ayan. Now, sphingomyelin can be, as I said, no, can be found in large quantities in your brain and nerve tissues, no? And a genetic defect, no, which results in lack of your sphingomyelinase, no, results in the accumulation of sphingomyelin. Okay? Ayan. So remember, a defect, no, in sphingomyelinase, no, ibig sabihin hindi gumagana yung kanyang sphingomyelinase, there will be an accumulation of sphingomyelin. And this disease is known as Niemann-Pick disease, no? Ayan. So Niemann-Pick disease. Yan. Now, you may wonder, sir, paano naging problema yung yung maraming sphingomyelin? Eh, sabi mo kanina, yung nerves, 'di ba may covered siya na sphingomyelin. And because of the sphingomyelin, mabilis yung signal transduction. Okay? Ayan. 'Di ba yun yung sinabi natin kanina, no? Eh ano nga, pag dumami yung sphingomyelin, bakit siya masama, no? Kasi you can never turn off your signal transduction. Forever kang excited, no? Forever kang mayroong muscle contractions, no? Ayan. So laging mayroong ganon. So Niemann-Pick disease 'yon. All right? Or sobrang sensitive ng iyong nerves, okay? Yan, that's your Niemann-Pick disease. All right? Yan a problem, no? A defect in sphingomyelinase. Next, sphingolipid is your glycosphingolipid. So glycosphingolipid can either be a cerebroside or a sulfatide. Okay? Now, yung glycosphingolipid, no, it is just ceramide with one or more sugars, no? Medyo madali lang tandaan, no? Kasi sabi natin kanina, no, ceramide is simply sphingosine plus a fatty acid. Dagdagan mo ng sugar, the glyco is sphingolipid. All right? Ayan. Now, galactosylceramide can be found in the brain, no, and other nervous tissue. So galactose, yung sugar in the brain and nerve tissues. Pero glucose, yung sugar na nandoon sa extraneural tissues. All right? Ayan. So again, galactose, brain, glucose, other tissues. Okay? Now, you have also, no, these other types of your glycosphingolipids. You have your cerebrosides. Ayan. So, cerebrosides are simply ceramide monohexosidases. Okay? Or ceramide monohexosides. Ayan, mali na naman ako ng basa. Ayan. Or hexosyl ceramide. So, ibig sabihin, cerebrosides are simply ceramides plus a 6-carbon sugar. Okay? Para madaling tandaan. Ceramide plus 6-carbon sugar, that's your cerebroside. Okay? Now, there are two diseases, no, that are linked to problems, no, in enzymes that degrade this cerebroside. So you have the Gaucher's disease and you have the Krabs disease. Okay? So para madaling tandaan, ganito yung lagi ko sinasabi, galactocerebroside, no? Saan nga ulit yung galactocerebroside? Galactocerebroside or galactosylceramide can be found in the brain and nervous tissues, no? So yung galacto, may K yan, Krabs disease. Okay? Ayan, so di ba? Galacto, ayan, may K, Krabs disease. Okay? And then, ang glucocerebrosidase, yung problem naman, gluco, may O, may U, no? Edi, Gaucher's disease, no? Ayan, so glucocerebrosidase, no? Is to Gaucher's disease, and then galactocerebrosidase is to Krabs disease. Okay? Yeah, so remember that these are defects in these enzymes. Okay, yeah, so they have difficulty, no, removing, no, these cerebrosides. And then we have the sulfatide, ayan, which is present in high amounts also in the myelin sheath. Ayan. And then lastly, we have ganglioside. Ayan. So yung ganglioside, ang tandaan nyo dito ay, it is a glucosylceramide, no, with sialic acid. Okay? Now, the principal sialic acid in ganglioside is your neuraminic acid. Okay? Ayan. So, kung meron kayong dapat tandaan, no, yun na lang. No? So, ganglioside, sialic acid. Okay? Ayan. And then, yung most common sialic acid is neuraminic acid. Okay? It is found in human tissues. And yung problem naman ng accumulation of gangliosides in the brain ay tinatawag na Tay-Sachs disease. Alright? Ayan, so, i-summarize natin, no? Ayan, so, kapag meron tayong problem in sphingomyelinase, no, that is your Niemann-Pick disease, no? Ayan. So pag ang problem is beta glucocerebrosidase, that's your Gaucher's disease, okay? Kapag galactocerebrosidase, no? K, Krabs disease, okay? Tapos kapag Tay-Sachs disease naman, this is your sialic acid, no? Gangliosidase, no? Ayan, there's accumulation of gangliosides, no? Tay-Sachs disease. Alright? Ayan, so that basically sums up, no, yung ating phospholipids, no? We will now proceed to another group of lipids, no? We call the steroids. Ayan. Normally, yung steroids, hindi ko talaga masyadong dinidiscuss, no, in biochemistry because this is more a PCOG, pharmacognosy stuff. Pero I will point out the most important things, no, when it comes to steroids. Okay? So remember that steroids, no, steroids have the CPPP moiety, no? Ayan, CPPP moiety. Ayan. So familiar with the CPPP? This is your walang kamatayan, cyclopentano perhydro phenanthrene, no? Ayan, hydrophenanthrene moiety. Alright? Now, why is this called cyclopentano perhydrophenanthrene? Kasi this is the phenanthrene moiety. Ayan, tatlo siyang aromatic ring. Ayan, so this is your phenanthrene. Ayan, okay? Tapos, kakabitan natin ng cyclopentane yung structure. Okay? Diba? Ayan, cyclopentane. 1, 2, 3, 4, 5. No? Ayan. So, this is your cyclopentano tapos phenanthrene. Okay? Now, if I remove all the double bonds in your phenanthrene, no? Ayan, burahin ko lahat ng double bonds in phenanthrene. Ayan, burahin ko to. Ito. That now becomes your cyclopentano perhydrophenanthrene. Okay? Yeah, so remember that yung term na perhydro, it means walang double bonds, no, in the structure. Okay? Ayan. So the basic skeleton of steroids is your cyclopentano perhydrophenanthrene. All right? Now, This is the structure of your cholesterol. Ayan. Cholesterol is the most widely occurring sterol. Okay. And it is the precursor to digestive and solubilizing agents such as the bile acids. Ayan. So remember that bile acids are synthesized in your body, no, in order to metabolize, no, the fats in your diet. Okay? So yun yung ang purpose ng bile acids. In fact, bile acids are also steroids in nature. Okay? So, cholesterol na dapat nyo natandaan dyan is the most widely occurring steroid. Ayan. Now, next we have the other steroids, no? So for example, wag niyo nang pansinin tong coprostanol, ito na lang, ergosterol, okay? Ergosterol, ergo, no? Pag nakakita tayo ng prefix na ergo, ibig sabihin fungus, no? Ayan, so galing sa fungus, no? Parang ergot alkaloid, 'di ba? Galing sa fungus 'yon, okay? Yeah, so ergosterol present in fungi, but plants also have ergosterol, okay? Now, what is important in ergosterol is that it is the important precursor of vitamin B2. Okay, yan. So, it's a precursor of vitamin B2. Ayan. So, ergosterol. Yung precursor to vitamin D3 ay tinatawag nating cholecalciferol. Okay, ayan. So, cholecalciferol, sulat natin, cholecalciferol. Yan, this is your pro-vitamin B3. All right. And then another steroid, no, is your beta-cetosterol, which is the most common sterol in plants. Okay? Kung sa mammalian tissues, it's your cholesterol. Sa plants, it's your beta-cetosterol. Okay? Next, we have your bile acids. Now, the bile acids are synthesized in the liver from cholesterol. Ayan. And ito yung mga different types of bile acids, no? Like your cholic acid, desoxycholic acid, and lithocholic acid. Ayan. Now, the synthesis of steroids is quite lengthy. Okay, ayan, mahaba-haba ito. But remember, ayan, balikan ko lang yung fatty acids, no? Although medyo nadaanan ko siya kanina. What is important for you to remember regarding the biosynthesis of fatty acids is that it comes from the acetate pathway, no? Ayan, so acetate. Ang precursor of your fatty acids ay acetyl-CoA. All right? Ayan, so acetyl-CoA. For the steroids naman, no? Ang pathway na dinadaanan niya is the acetate mevalonate pathway. Okay? Yan. So, dalawa na yon. Acetate mevalonate pathway. And yung fatty acids, no? Kung yung fatty acids require only, no? Acetyl-CoA, isa lang, no? Every time humahaba yung kanyang chain, okay? For the steroids, kailangan niya ng tatlong acetyl-CoA to produce the mevalonic acid. Alright? Ayan. So, kaya siya tinawag na acetate mevalonate pathway kasi mag-start siya sa acetate pathway, no? Acetyl-CoA enzyme A pa rin, yung precursor of your steroids. Then, magko-combine yung tatlong molecules of acetyl-CoA to form your mevalonic acid, no? Ito 'yon. Ayan. Okay? So, mevalonic acid yung na-form niya. Kaya tinawag siyang acetate mevalonate. Alright? Now, what is important here is that HMG-CoA reductase is a very important enzyme in the synthesis of your steroids. Okay? Yeah, so HMG-CoA reductase. Okay? So, ano ibig sabihin ng HMG? Yan, this is your hydroxymethyl glutarate. No? Ayan, hydroxymethyl glutarate. Glutarate, no? Ayan. So yan yung ating HMG, no? So ito siya. HMG-CoA hydroxymethyl glutarate coenzyme A, no? HMG-CoA. Now, HMG-CoA reductase, no, is an important enzyme in the synthesis of your steroids, no? In fact, inhibiting the activity of your HMG-CoA reductase leads to lowered cholesterol level, no? Ayan, so sinong mga compounds yung ginagamit natin as HMG-CoA reductase inhibitors? See? Correct. Statins. Ayan. So, perfect. Yeah, so remember na statins are inhibitors of HMG-CoA reductase, no? Ayan, kasi sila ay anti-hyperlipidemics or anti-hypercholesterolemics, no? Ayan, so yun yung purpose nila. Alright? Ayan, so yun lang naman yung important dito, no? But just to run through the synthesis, ay shucks, hindi nyo pala nakikita. Sorry sa mga nasa physical world, yung mga nasa online world, kitang-kita nila yung mga pinagsasabi ko. Ayan, so ito yung ano, no, acetate mevalonate pathway, hindi pa rin makita. Okay, ayan. So, hindi pa rin makita. Ayan. So, anyway, lahat ng sinabi ko kanina, yun din yun. So, sa acetate mevalonate pathway, 3 molecules ang acetyl-CoA. Yun lang yung dapat yung tandaan, no? So, 3 agad na molecules ng acetyl-CoA magko-combine, no, through Claisen condensation, a Claisen reaction, and that will produce your, ayan, ito, HMG-CoA. Ito, ayan. So, nakikita ba yung HMG-CoA? Ayan. Ayan, so, ayan, so siya yan, no? HMG-CoA, tapos HMG-CoA reductase is needed in order to produce your mevalonic acid. Ayan, MVA. Ayan. Sige. Now, habang nandito pala tayo, meron pa akong isa pang gustong sabihin, no? Tungkol sa mga fatty acids na hindi pa natin na-mention. Para madali lang mag-derive, no, ng mga pangalan. Okay. So, are you familiar with the dicarboxylic acids? Dicarboxylic acids? And yes, no? Yes. Yan, gusto ko pag may narinig akong yes. Yan, dicarboxylic acids. So, kanina, di ba, sinulat na natin yung mga saturated and unsaturated fatty acids. Ngayon naman, pag-usapan natin yung mga dicarboxylic acids. Okay? Ayan. So, are you familiar doon sa code natin? Yung, oh my, such good apple pie. Sweet as sugar. Ayan. Okay? Ayan. So yan yung ano, dicarboxylic acids na code naman natin, no? Kaya tutunan ko to doon sa teacher namin ng organic chemistry before. Kaya, oh my, such good apple pie, sweet as sugar. Ayan. Okay? Ayan. So, hanap ako ng kulay. Ayan. So, this is your carbon 2. Sige. Carbon, hindi. Carbon 2. Ayan, so 2 and then plus 1 plus 1 plus 1 lang. No? So 2, 3, 4, 5, 6, 7, 8, 9, and 10. Okay? Ayan, so basically yan yon. So yung O, this is your oxalic acid. No? Ayan, oxalic acid. Yung structure ng oxalic acid mo, COOH, and then COOH ulit. No? Ayan. Okay? Tapos M, this is your malonic acid. Ayan. So magdadagdag ka lang ng isang carbon in the middle, CH2. Ayan. COOH. Ganon din dito, COOH. No? Ayan. Tapos, S is your succinic acid. Succinic acid. This is your glutaric acid. Tapos this is your adipic acid. Pimelic acid. Azelaic acid. Nakalimutan ko yung isa. Ayan. Hindi ko maalala kung sino dito yung suberic, no? Ayan. So, nakalimutan ko. I'll get back to you on that. Pero parang hindi ako magkamali, basta ito na muna. Ayan, okay. So, we have, oh my, such good apple pie, sweet as sugar. So, oxalic, malonic, succinic, glutaric, no? Ayan, hanggang C5. Ayan, punta tayo dito. Yan, di ba yung hydroxy methyl glutaric acid, no? Yan, dito lang din yon, no? So, glutaric acid, so C5, no? So, isipin niyo yung structure ng glutaric acid. Oo, so C8 suberic acid. Ayan, thank you for the assist. Ayan, nangyari. Okay, this is suberic acid. Nalimutan ko yung last na S actually. Ayan, so suberic acid. Ayan. Alright, sige, laktawan naman natin yan. So C5, no? So you have your COOH. Ayan, tapos CH2, CH2, CH2. Tapos COOH ulit. Alright? Ayan. So, pag sinabing beta hydroxy, no? Beta hydroxy methyl glutaric acid, no? So you just have to add, no, the groups in your glutaric acid. All right? Ayan. So kaya ko lang naalala. Ayan, itong dicarboxylic acids. Ayan, oh my such, ayan, sebacic. Ayan. So thank you for that, no? Sebacic acid yung C10. Ayan. Ayan. So, yung mga ano to, yung mga unplanned things. Okay, sebacic. Ayan. Okay? Ay, question? Akala ko, ay, alright. So, ulitin natin, oxalic acid, malonic, succinic, glutaric, adipic, pimelic, suberic, azelaic, and sebacic acid. No? Ayan. Ano lang yan? Added info lang. Kasi, di ba, minsan nakakalimutan na natin yung mga, ano, yung mga ganyang bagay. Alright. So, next is, yan, balik tayo dito. Yeah, kasi medyo malabo kasi, no? Malabo yung structures, hindi nakikita ng mga nasa physical world. Ayan. Pero yun lang naman yung dapat yung tandaan. Yung HMG-CoA, no? Yung precursors. Ayan. So, that's basically it. All right. So, this is, no, another way of looking at the synthesis of your steroids. Ayan. Okay, so for the synthesis of the steroids, yeah, you have your acetyl-CoA, hindi pa rin nakikita. Ayan, acetyl-CoA, which combines with another acetyl-CoA to form acetyl-CoA, which combines with another acetyl-CoA to form your 3-hydroxy-3-methyl-glutaryl-CoA or hydroxymethyl-glutaryl-CoA, which becomes your mevalonic acid. Tapos, in the end, no, you have these two. Then you have your dimethyl allyl diphosphate at saka your isopentenyl pyrophosphate. Ayan, burahin lang natin to. Ayan. So now, itong dalawa, si dimethyl allyl diphosphate, ayan, dimethyl allyl diphosphate and your isopentenyl diphosphate, these two, no, are the immediate precursors of the terpenes. Okay, ayan. So, tandaan din to. These two, your isopentenyl pyrophosphate and your dimethyl allyl diphosphate, okay, they are the precursors of the terpenes, no? Ayan. So, the acetate mevalonate pathway kasi is also used, no, in the synthesis of your terpenes. Okay? Ayan. So, they have this isoprene unit. Okay, so dimethyl, diallyl, diphosphate, and isopentenyl pyrophosphate. All right. Terpenes. Ayan, they form your terpenes. The steroids are terpenes. No? Ayan, terpenoidal in structure din sila. Ayan, so these are just more structures, no? Ayan, pero siguro laktawan na lang natin to. At na-disconnect po tayo ulit. Yeah, so next group of compounds, no, aside from your steroids, ay tinatawag na eicosanoids, okay? Yeah, so what are these eicosanoids, no? So eicosanoids come from your arachidonic acid, okay? Ayan. So remember, si arachidic acid ay C20 saturated fatty acid. Si arachidonic acid ay C20 unsaturated fatty acid, okay? And your arachidonic acid, no, ay yung precursor of your eicosanoids. They are formed in the body from essential fatty acids such as your linoleic and alpha-linolenic acid. Now, these eicosanoids can be active at very low concentrations. In fact, they are hormone-like. Okay? Yan, examples of your eicosanoids ay yung prostaglandins, your leukotrienes, your thromboxanes. Okay? Ayan. So they act, no, in the body like hormones, no, particularly as signals of pain, no, and inflammation. They can also regulate blood pressure, contractions of the smooth muscle, gastric secretion, and as well as platelet aggregation. Ayan. So nabanggit na natin to kanina, so move tayo dito. Ayan. So another terminology that is similar, no, dito sa eicosanoids ay yung term na prostanoid. So this is just an umbrella term, no? Ayan. So pag tinanong yung umbrella term, they are prostanoids, no? Kasi they are from prostanoic acid, okay? And the prostanoic acid has this cyclopentane ring, no? Yan, at carbon 7, and this one. Ayan. So yan yung mga prostanoids. Now, your eicosanoids are in fact prostanoids. Okay? Ayan. And as we mentioned before, they are your prostaglandins, thromboxanes, and leukotrienes. Ayan. Ayan. So, how are they formed in the body? Ayan. So, punta tayo dun sa synthesis nila in our body. Yeah, so remember that you have your arachidonic acid. Ayan. Yeah, so you have your, nakikita ba? Natakpan, no? You have your arachidonic acid. Oops. Ayan. Gawa natin. Ayan, so you have your arachidonic acid. Ito yung structure ng arachidonic acid. It's a C20 fatty acid. Hindi kasi pwedeng magsabay yung ano, pen at saka yung, alam ko na kung paano. Okay, teka. Wait. Annotate tayo. All right. So arachidonic acid, this is the primary precursor, no, of your eicosanoids. Okay? Now, meron tayong dalawang pathways. You have the cyclooxygenase pathway and the lipoxygenase pathway. Okay? So remember that in the cyclooxygenase pathway, this is where the prostaglandins are produced. No? Ito yung tinatawag ninyong COX. Ayan. Kaya nga, di ba, meron tayong tinatawag na COX-1 at saka COX-2 inhibitors. Okay? So remember that COX-1, no? COX-1, the cyclooxygenase 1, no, pathway leads to production of ubiquitous, ayan, prostaglandins. Okay? So when you say ubiquitous prostaglandins, these are prostaglandins that are produced normally by the body. Okay? And some of these prostaglandins are in fact beneficial to the body. They form the mucous membrane, no? They form the mucous, no? Protective lining, no? In the gastric mucosa. All right? Ayan. So, nape-prevent, no, yung ulceration in the stomach because of the production of these prostaglandins. Okay? They are ubiquitous. Okay? Now, yung COX-2 naman leads to prostaglandins which are required, no, as pain signals or inflammation signals. Okay? Ayan, na napoproduce lamang yung prostaglandins from cyclooxygenase 2, no, if there is a trauma, no, pag may wound, no, pag mayroong sakit, no, ayan, may mga invaders, no, may tissue damage and others, okay? Yun yung COX-2, okay? Now remember that initially, no, what we have, no, as analgesics and anti-inflammatory ay non-selective COX inhibitors. Ayan, di ba? Ito yung meron tayo, non-selective COX inhibitors. So ibig sabihin, they can inhibit both COX-1 and COX-2, okay? And because of that, no, nagli-lead ito sa gastric ulceration, okay? Dahil na-inhibit pati yung ubiquitous prostaglandins, no, which coat your gastric mucosa. All right? Now, later on, nagkaroon ng production, no, of selective COX-2 inhibitors. Kasi during inflammation, you just want to reduce the amount of the prostaglandins from COX-2. Okay, yun lang naman. Kaya gumawa sila ng selective COX-2 inhibitor, no? Yan. So this one includes yung celecoxib, no, rofecoxib, no? Ayan. So yun yung examples ng selective COX-2 inhibitors. All right? Yan. Now, compared to your prostaglandins, no, which directly come from arachidonic acid, okay, through the cyclooxygenase pathway, yung ating leukotrienes, no, yan, would actually come from your lipoxygenase pathway. Ito naman, okay? So the immediate precursor of your leukotrienes ay si HPETE, no? Ayan, so that is just your hydroperoxy eicosa tetraenoic acid, okay? So hydroperoxy eicosa, tapos baba na natin, tetraenoic acid. Ayan, so that's your 5-HPETE, no? So in the lipoxygenase pathway, the leukotrienes are produced, no? Ayan, so yun lang naman yung pinapakita in this particular diagram. Next, the thromboxanes and prostacyclines are also produced, no, via the cyclooxygenase pathway, no, but in a different pathway compared to your prostaglandins, no. So ang common na tinatanong dito ay which of the following eicosanoids are produced, no, let's say from HPETE, no, ayan. So dapat alam, kunwari ang choices ay thromboxanes, no, prostacyclines, prostaglandins, leukotrienes. The answer is leukotrienes. Alright? Ayan. So, yun lang naman yung important over here. Ayan. So, that is your lipids, no, and related compounds. Okay? So, any questions so far? Ayan. So, tatalon tayo ulit. Ayan. Pupunta tayo sa nucleic acids. Ayan. Puro lang muna tayo structures ngayon. All right. So now we are in nucleic acid, nucleotides and nucleic acids. Ayan. Okay, so carry pa. Ayan. So mga 2:45 pwede tayo mag-break. 15-minute break. Ayan, para saktong 3 balik tayo. All right. So for nucleotides and nucleic acids, ganun pa rin naman, no? Structure, function, biosynthesis, and then later on metabolism, no? So let's begin with your nucleotides, no? Ayan, nabanggit na natin to kanina. So review na lang din. Well, this is a review, pero review of the review. Ayan. So your nucleotides are composed of 3 components. You have the sugar, which can be ribose or the oxyribose. You have the phosphate, no? And you have the base, no? Sa nitrogenous base, you have the purines and the pyrimidines, no? Yung purines, these are your adenine and guanine. And then sa pyrimidines are your cytosine, no, uracil, and thymine. Okay? Now, take note na when I remove, no, when I remove the phosphate group, no, the phosphate group in the molecule, okay? Instead of calling it a nucleotide, it now becomes a nucleoside. Okay, yan. So, pag binawasan natin, no, ng phosphate group, yung ating nucleotide, ayan, what you have is a nucleoside. That is why, no, we call yung mga, let's say, AMP, no, GMP as adenosine monophosphate, no, ayan. Guanosine monophosphate. Guanosine and adenosine are names for nucleosides. Okay? Ayan. So mamaya, no, tingnan natin yung naming ulit nila. Now, these are the structures of your purines and your pyrimidines. Okay? Ayan. So, be familiar with their structures. Ayan. So, ito lang yung madalas na iniisip ko, no? Ayan. Yung mas mahabang pangalan, pyrimidines, no, have a small structure. Okay? Yung mas mahabang pangalan, pyrimidines, is smaller yung structure. Yung mas maiksi yung pangalan, siya yung mas malaki yung structure. There are two cyclic rings, no, in your purines. Di ba? Ang dali lang tandaan. Meron pang isa, no? Ayan, so sabi nila, no? Na-disconnect na naman po ako. Na-disconnect ako dito, pero nandun pa rin ako. Okay. Sige, habang nagde-decide siya kung kukonect siya o hindi. Ayan, okay. Perfect. Ayan, so remember this code, no? Ayan, so may code ako nakita diyan. Ayan, so sabi nila, no, adenine. Adenine, ang pangit, teka lang. Ando natin 'to. Ayan, so remember this code, Ayan, teka lang, sulat natin. So adenine and guanine are pure and uncut. Ayan, okay? Ayan, so kung hindi nyo masyadong bet yung sinabi ko kanina na pag mahaba yung pangalan, maliit yung structure, one ring. Pag maiksi yung pangalan, malaki yung structure, two rings, no? Just remember, adenine and guanine are pure, uncut, okay? So explain natin tong code na ito. You have here adenine and guanine, okay? So yung adenine, nakasulat pala dito sa baba. So adenine and guanine, they are purines. Ayan, they are purines. Okay? Yan, and then they are uncut, no? Hindi sila, no? They are not cytosine, uracil, and thymine, which are pyrimidines. Okay? Ayan. So, yan, no? Para madaling tandaan. All right? Now, let's describe the structure of your adenine and your guanine. Now, take note that your adenine has the amine group at carbon number 6, no? Ayan, may amine group siya at carbon number 6. Ayan. While your guanine has the ketone group and the amine group, no? Ayan. So, more than one functional group yung added to the structure of your guanine. Again, yung adenine, you first draw the structure of the purine, okay? Yeah, so ito kasi yung structure ng purine. Kung magdodrawing kayo ng purine, ganito lang yon. Ayan, tapos maglagay kayo ng nitrogen dito. Nakita ba? Ayan. So, N, tapos maglagay tayo ng double bonds, double bond, double bond, double bond, double bond. Ayan. So, yan yung structure ng purine. Alright? So, for your adenine, burahin muna natin. Pagandahin natin yung drawing. So, 1, 2, 3, 4, 5, 6. Yan. Tapos, 5. Tapos nitrogen here, nitrogen here, nitrogen here. Na-disconnect na naman po ako. Alright. Ayan. Tapos double bond here. H dito. Tapos double bond, double bond, double bond. Aha. Okay. Okay na siya. Yeah. So this is your purine. Okay. So for your adenine, you just have to add an amine group over here. Yun na yun. Okay? That's your adenine. Okay? Ayan. Gets? Tapos si guanine, ayan, magbura tayo. Si guanine, yan, burahin mo yan. Maglalagay tayo ngayon ng double bond O, ketone group over here, and then over here, amine group. Kaso, magbabago ngayon yung bonds dito sa loob. We have to erase some of these. Ayan, so, kailangan natin tanggalin ito. No? Ayan. So, that is now your guanine. Okay? Di ba? Madali lang. Ayan. 'Di ba? Kaya na maging familiar with the structure. Okay? Ayan. Napapailing sila eh, no? Ayan. So, para madali, isa pang madaling para isa pang memory, memory cue, no? Adenine is amine. Okay? Ayan. So, ganun lang isipin nyo. Adenine is amine. Okay? So, 'di ba? So, adenine has the amine group. Ayun. Okay? So, bakit ko paulit-ulit na sinasabi ito? Kasi lumalabas talaga yung structures, no? In the exams, okay? So, adenine is amine, okay? Tapos, si guanine, okay, ay meron siya nito, no? Ayan. This looks like the guanidine group. Ayan. So, that's the guanidine group, which you will again see in an amino acid, no? Kay arginine, okay? Yeah, so that's the guanidine group. Tatlong N, okay? Yeah, so imagine niyo para siyang yung thumb spinner. Ano ba tawag doon? Fidget spinner. Ayan, so thumb spinner, Para siyang fidget spinner, 'di ba? Yung itsura niya, may tatlong N, na? Ayan, that's your guanine. Okay? Punta naman tayo ngayon doon sa pyrimidines. Okay? Yan. So, pag sinabing pyrimidine kasi, yan, burahin ko muna ito lahat. Okay, so, when you say pyrimidine, actually gusto ko na magsulat doon, kaso hindi makikita ng ating mga friends from online world. Ayan, so, this is your pyrimidine. Ayan, so, nitrogen, nitrogen, tapos maglagay ka lang ng double bond dyan. Okay, that is now your pyrimidine. Okay? Ayan, so, okay, balance naman. For cytosine, so for cytosine, you add here NH2, and then there is a C double bond O over here. No? Of course, dahil nagdagdag ako ng C double bond O, magtatanggal ako ng double bond. Ayan. Okay? That's it. That is already my cytosine. So, syempre, dadagdag ako ng H kasi nagtanggal ako ng double bond. Okay? Yeah, so this is your cytosine. Okay? Paano madaling matatandaan yung cytosine? The cytosine just looks like an inverted guanine. Okay? Yan. So, pansinin nyo to. Ito yung amine, ito yun. Okay? Tapos, ito yung C double bond O, ito yun. Ayan. Di ba? Parang inikot mo lang. Okay? Yan. Why? Kasi these two are actually complementary. Okay? Which we'll talk about in a little while. Okay? They are complementary. Alright? Yeah, so para lang siyang binaliktad na guanine. Alright? That is your cytosine. Now, yung uracil, ayan, so pagdating naman kay uracil, ibalik lang natin yung structure. Ayan, so drawing natin ulit yung pyrimidine. So the pyrimidine is ganito, ganito, double bond dito, double bond dyan, double bond dyan. Ayan. So yung uracil, you just add C double bond O over here, C double bond O over here, tanggalin natin yung mga double bonds. Yan, tanggalin mo to, tanggalin mo to. Then we add hydrogens over here, and then over here, no? That is now your uracil. Okay? Ayan, uracil. Nasundan yung pag-drawing natin. Yan. So, drawing nyo muna yung pyrimidine. Sige, ulitin ko na lang sa board. Okay? Tapos, dagdagan natin ang methyl group dito. That is now your thymine. Okay? Yan. So, for the benefit naman of those who are here, no, in the physical world, ulitin natin. Okay. Okay. Ay, wala pala. Hello, hello. Ayan. Okay, so first is, unahin natin si purine. Ayan. So, remember si purine, maglalagay ka lang ng nitrogen dyan. Tapos nitrogen dito, nitrogen dito. Alright? Ayan. Okay? Tapos double bond dito, tapos double bond dito, double bond dyan, double bond dyan. Okay? That is now your, dagdagan natin ang H dito. That is now your purine. No? Ayan. So that's the basic structure of a purine. Okay? Now for adenine, okay, all you have to do is add an NH2, an amine. Okay? At carbon number 6. Ayan. So NH2. Wala siya masyadong magagalaw ng mga double bonds. Okay? Yan pa rin yun. So, yan na yun. That is already your adenine. Diba? Yan, dali lang, no? Yan, now, so, si guanine, i-rewrite natin siya para mas madali. And kaso ang pangit ng drawing ko talaga. Ayan. Now, for guanine, so for guanine, ano yung dapat nating tandaan? May NH2 dito. Tapos may double bond O dito. Okay, this is your guanine. Kaso, naglagay ako ng double bond dito, 5 na yung bonds ng carbon. Okay? Ayan. So actually, I am also a teacher of organic chemistry, no? This is one of my pet peeves, no? Ayan. So, carbon na may 5 bonds. Okay? O kaya hydrogen na mayroong 2 or more na bonds. Ayan. Asar na asar talaga ako doon, no? Ayan. Imaginein mo, kunwari, H tapos may ganun pang H, no? Ayan. Ang daming bonds, no? Ayan. So, dinaig pa si Oppenheimer, no? Ayan. So, hindi sasabog yung mga yan. Alright. So, tanggalin mo to. Ayan. Since nagtanggal ako dito, I will now add another hydrogen here. That's now your adenine. Okay? So this is your adenine. That's now your guanine. Ayan. So this is your adenine. Ayan. Tumalon kasi utak ko eh. Ayan. Okay? Ayan. So di ba madali lang tandaan? Okay. So next is drawing tayo ng pyrimidines. Okay? So for the pyrimidines, ayan. So N, maglagay ka lang ng N doon. So, para mas maganda, burahin natin. So, N. So, yung mga nasa online world, chillax lang kayo dyan. Ayan, kasi drinawin ko na siya sa inyo. Hindi lang nakita ng mga nandito sa physical world. Ayan. So, this is your pyrimidine. Okay, yan, pyrimidine. This is the basic structure of pyrimidine. Now, for your cytosine, okay, sabi natin, kamukha niya yung guanine, balik pa rin mo yung dalawa. Okay? So, you place here the double bond O, tapos lagay mo dito yung NH2. Okay? Ayan. So, wala akong gagalawin dyan. Ito gagalawin ko kasi sumobra na ng bonds yung carbon. Dagdag ko na lang siya ng hydrogen. That is now your cytosine. Diba? Dali lang, no? Okay. So, next. Drawing natin yung dalawang magkapatid, no? Si uracil at saka si thymine. Ayan. Yan. Sabi sa inyo, huwag kayo mag-memorize. Be familiar. Yan. Okay. So, 1, 2, 3. 1, 2, 3. Ayan. Okay? Ayan. Now, for your uracil, maglagay ka ng double bond O dito and then another double bond O dyan. Okay? Para siyang ganon. Okay? Yan. So, para siyang palaka, no? Nakaganon. Ayan. So, yan, ito yung kamay niya, oh, ayan. Diba? Ang cute. Yan, tapos burahin mo ngayon yung double bonds na sumobra. Okay? Add hydrogens, kasi yung hydrogen dapat 3 yung bond. Okay? 1, 2, 3, 3. Okay? Hydrogen, 1, 2, 3. Okay? Yun yung ginagawa natin, no? Kaya tayo nagdadagdag ng hydrogen. Kailangan tatlo yung bonds ng hydrogen. Alright? Yan. So now, you have here your uracil. Ayan, di ba? Okay, next. Gawin natin si thymine. Sabi natin, si thymine, uracil lang yan. Yan, dinagdagan natin ng methyl group dito. Okay? Yeah, so tanggalin natin double bond, hydrogen, hydrogen. This is now your thymine. Okay? Ayan. So, yan. That's how you can remember to draw the structures of your bases. Okay? Ayan. Kasi medyo malabo, no? Hindi masyado nakita on screen. Kaya nag-drawing na lang ako ng hiwalay for you. Alright? Okay? Siguro one last slide and then let's have a break. No? Ayan. Kasi baka na-overwhelm kayo. Ano ba yung binibilang-bilang na yan? 5-5, 3-3, no? Ayan. May double bond dito, tapos dinagdagan bigla ng hydrogen to, no? Ayan. So, pero clear ba tayo doon? So, kaya nang mag-drawing ng structure. Okay? Ayan. At least, kaya mag-recognize ng structure. Kaya, no? Ayan. So, tandaan nyo lang yung mga sinasabi ko, no? Adenine, amine. Okay? Ayan. Tapos, guanine, may guanidino group, no? So, hindi ko pala hina-highlight yung guanidino group. The guanidino group din, hina-highlight ko siya dito sa online world. So, sa physical world naman tayo. Ayan. Yung guanidino group ay ito. Ayan. Yung parang fidget spinner. Thank you for that term, no? Ayan. Bilang isang millennial, hindi ko alam yon. Okay. So, fidget spinner, no? Ayan. So, yan yung guanine. May guanidino group. Tapos, your cytosine, your uracil, and your thymine, no? So, yung uracil and thymine, pareho lang. Ang pinagkaiba nila yung methyl group, no? Ayan. Merong methyl group na additional si thymine. Alright? As para siya nakaplakdang gano'n. Gano'n, no? Ayan. So, ang cute kaya, no? Pag i-imagine ko siya, laging gano'n yung naisip ko, no? May nakadapa, no? Isipin nyo na kung tao yan or palaka, no? Ayan. It's up to you. Last slide before we have our break. The two sugars, no, that are present in your nucleic acids are your ribose and your deoxyribose. Now remember that these two are your pentoses, no? Ayan. So these are pentoses. They come from your pentose phosphate pathway. All right? Ayan. So yun yung dapat niyo pang tandaan, no? These sugars, ribose and deoxyribose are from pentose phosphate pathway, no? Ayan. And now, deoxyribose, no, is called deoxyribose because walang hydroxyl group at position number 2, no? Ayan. Hindi ko alam ba't ako nag-drawing dito. Kasi kita naman siya dito sa online world. Ayan, so take note na ito lang yung difference nila, no? So the deoxyribose doesn't have the hydroxyl group at carbon number 2. All right? Yeah, but both are pentoses, both are riboses, no? Ayan, yun lang talaga yung difference nila. All right? Ayan. So ano muna tayo? Chill muna tayo, no? So let's have possibly a 10-minute break. Ayan. Kaya ba? Or 15, sige. Mga 15-minute break. Gusto ko sana 10 lang. Ayan, pero baka malayo-layo pa yung pupuntahan ninyo. Ayan, so let's have a 15-minute break. Ayan. So when you come back, no, nucleotides, naming nucleotides, and then ano na tayo? Synthesis, no? Synthesis of your nucleotides, no? Ayan. All right. So yun. Okay. So, remember na, ano nga ba yun? May iba akong inisip. Okay, so remember na, the DNA, no, has two strands. Okay? So, if one is in the 5' to 3' direction, then the other strand will be in the 3' to 5' direction, no? Ito yung tinatawag natin na anti-parallel. Ayan, anti-parallel orientation uh, of the strands of your DNA. The same is true for the RNA, no? If the RNA would have a double strand, no, then it would also be anti-parallel. Okay? Yan. Now, when we talk about the structure of your nucleic acids, yan, meron tayo four levels. Primary structure, secondary structure, tertiary structure, and quaternary structure. Okay? So, ano ba ito mga ito? Pag sinabi natin primary structure, it's basically the sequence of bases. Okay? So, primary structure, it's the sequence of bases. Okay? From 5' to 3'. Okay? So, again, by convention, lagi tayong susulat ng primary sequence. Okay? So, tandaan na, lagi tayong nagsusulat ng primary sequence 5' to 3' by convention. Okay? Anong ibig sabihin nun? If I would write, let's say, 5', G-A, T-A, C-A-A, yan, to 3'. Yan yon. Okay? That's the primary sequence. Okay? Now, sometimes, hindi natin sinusulat yung 5', yung 3'. Ganito lang natin siya sinusulat. Ayan. Now, it's understood na the ends are actually 5' over here and 3', ayan, over here. Okay? Ayan. So, pag hindi nyo nakita yung 5', 3', understood yon, no? Huwag nyo nang tanong, no? Kunwari sa exam, huwag nyo nang tanungin sa examiner. Ma'am, sir, excuse me po, nasan po yung 5'? Ayan, hindi po naka-indicate, no? Ayan. Baka batuhin kayo ng batuhin kayo ng shock. Ayan, so 5' to 3' palagi, no? Pag nagsusulat tayo ng primary sequence. Alright. So next is the secondary sequence. For the secondary sequence, ayan, or for the secondary structure, this is the stable repeating conformational pattern in a polymer. Okay? So for your nucleic acids, it's the duplex, no, that is formed when two strands of the DNA or RNA combine. Okay? That's the secondary structure. Okay? Now, remember that what contributes to the secondary structure of the nucleic acid ay yung tinatawag nating base pairing. No? Ito yung complementary base pairing na tinatawag. Complementary base pairing. Now, the complementary base pairing between bases, no, was actually discovered by Erwin Chargaff, no? Ayan. So si Erwin Chargaff, yung naka-discover noon. Ayan, si Erwin Chargaff ng Chargaff's rule. Ayan. So Erwin Chargaff, no, actually mentioned na when you have a duplex DNA, no, when you have a duplex DNA, and then you determine the ratio, no, the ratio of purines and pyrimidines, it's always equal, no. And ang laging equal ay yung adenine kay thymine at si guanine kay cytosine. Okay, so he discovered that there is 1:1 ratio between adenine, thymine, and guanine and cytosine. No, yan yung sinabi ni Erwin Chargaff. Later on, no, through the two scientists, ito, mga wala ka kwenta-kwentang scientist, no, si James Watson, meron akong bias against these two persons, James Watson and Francis Crick. Ayan. Ayan. So James Watson and Francis Crick, later on, no, got a hold of this information from Erwin Chargaff. Tapos they finally published, no, they published the double helical structure of the DNA, no? Ayan. So take note na yung ginamit kong term ay published the double helical structure of the DNA, no? Ayan, kasi gusto kong bigyan lang, no? Ayan, bigyan ng tawag ito, justice, no? Gusto kong bigyan ng justice si Rosalind Franklin. Ayan. So another scientist. Yan. So basta may kinalaman sa DNA, ito yung mga dapat ninyong tandaan, no? Si Rosalind Franklin. Ayan. So, gusto ko siyang bigyan ng ng highlight. Ayan. Rosalind Franklin was actually the scientist who discovered, the structure of the DNA through her photograph 51. Ayan. So, ang tawag dun sa photograph niya ay si photograph 51. So, ang ibig sabihin ng photograph 51, yan yung pang 51st photograph na ginawa niya of the DNA. Okay? Now, Rosalind Franklin was working on an instrumental technique called X-ray crystallography. Ayan. X-ray crystallography. So basically, she used X-ray, okay, to determine, the bonding patterns of the bases in a DNA molecule. Okay? Dahil pang 51st photograph niya na ito, imagine niyo na lang gaano siya katagal nag-work, okay, sa laboratory to get this structure right. Okay? Ayan. So Rosalind Franklin, did a lot of experiments on the B conformation of the DNA. It's the physiologic form of the DNA, the B form of the DNA, and she was able to produce the photograph 51. Okay. Now, from the photograph 51, she was able to identify that the backbone of the DNA are at the outermost portion of the molecule. Sila ay nasa labas, wala sa loob. Kasi initial reports, the DNA structure during that time ay nagsasabing nasa loob daw yung sugar phosphate, ng DNA. According to Rosalind Franklin, siya na nagsabi in her presentation, in one of her lectures, that the sugar phosphate are in the backbone, nasa gilid, okay, nasa labas, okay. And according din kay Rosalind Franklin, because she saw the regular structure of the photograph 51, ang ganda, It's one of the most beautiful picture that you will see, of a crystal, of an X-ray crystallogram. Isa yun sa pinakamaganda, Napakaganda niya, So actually, from that photo, Rosalind Franklin also found out that the bases are in the middle, okay? They are in the middle of a double helix, okay? They are in a helical fashion, okay? It's consistent, Consistent yung turns of the helix, ayan. The bases, have a regular, okay, size, okay? Both horizontally and vertically, okay? So anong ibig sabihin ng both horizontally and vertically? Sabi ni Rosalind Franklin, pag mineasure ko yung diameter of the DNA, it's the same all throughout the molecule. Never siyang magbabulge. Okay? According din kay Rosalind Franklin, based on her photograph 51, the distances between these two bases, ayan, can also be calculated. Ayan, ito. They are also regular. Okay? According din kay Rosalind Franklin, the backbone of the DNA helix ay sugar phosphate, contrary to the belief during that time. Okay? Those were all her findings, Those are all her discoveries, which James Watson and Francis Crick, found out. Okay, yan na, Yan, so narinig nung dalawa, okay? Etong dalawang to may ambisyon, Ayan, so their ambition is to publish, the first, DNA structure. Okay? Kasi during that time, that was very important, What's the structure of the genome? Okay? Nag-uunahan sila, regarding this structure of the genome. So James Watson and Francis Crick talked with Erwin Chargaff. Erwin Chargaff validated the findings of Rosalind Franklin. 'Di ba sabi ni Rosalind Franklin, anywhere in the DNA molecule, walang bulge. The diameter is the same all throughout. How would that happen? It would only happen if you have a purine and a pyrimidine that always pairs up. 'Di ba? Yun lang yung ibig sabihin nun. Hindi pwedeng mag-pair up ang dalawang purines kasi liliit yung diameter. Hindi rin, lalaki yung diameter. Hindi rin pwedeng magsama yung dalawang pyrimidines kasi liliit yung diameter. So, dapat palaging purine at pyrimidine yung magkasama. Okay? So it was actually Erwin Chaga who mentioned na, tama yung hula ni Rosalind, no? Ayan. Tama, purine and pyrimidine yung lagi mag-partner. Kasi I found out, no, that there is 1:1 ratio between a purine, which is adenine, and thymine, which is a pyrimidine. The same is true for guanine, which is a purine, and thymine, which is a cytosine, which is a pyrimidine. Okay? Yan. So, cling. Okay, check. Check na dun sa dalawa, no? Ayan, check na. May next na kinausap si James Watson and si Francis Crick, si Linus Pauling. Ayan. Linus Pauling was working on alpha helical proteins. Okay? Ayan. So, he was working a lot on helixes. Okay? Ayan. Kinonfirm lang ni James Watson at ni Francis Crick kay Linus Pauling, tama ba that when you have an alpha helical protein, the X-ray crystallogram will look this beautiful? Parang ganon, no? So sabi ni Linus Pauling, ay oo, dami ko na nakitang ganon na proteins. Ang ganda. Ganda ng structure. Check na naman, no? Ayan. So, ngayon, ang ginawa nitong James Watson at Francis Crick, pumunta sa isa pang scientist, no? Idamay na natin siya. Si Maurice Wilkins. Ayan. So, Maurice Wilkins, ayan, is the boss of Rosalind Franklin. Okay? So, he's the boss, no, in the lab, no, where Rosalind Franklin worked. Okay? Ayan. During that time, si Rosalind Franklin was the only woman working in the lab. Okay? Di ba? Mag-isa lang siyang babae working in the lab. Then Maurice Wilkins gave the photograph 51 to James Watson and Francis Crick. Okay? And the rest is history. They published the findings of Rosalind Franklin. So, sino nagkaroon ng Nobel Prize? Yung dalawa, mandaraya, no? James Watson and Francis Crick, no? Ayan. So, kung ang tanong ay who got the Nobel Prize, no? In medicine and in physics because of their discovery of the DNA, James Watson and Francis Crick. Pero who discovered the DNA? Dapat si Rosalind Franklin. Okay? Ayan. Kasi she was the one, no, who first saw the photograph 51. And sinulat niya, no, yung findings niya, no? It's just that hindi niya pinublish, no, yung findings. Okay? Ayan. So, gusto ko lang i-highlight, no, because she's a woman scientist, which is very rare, no, until now. Okay? And hopefully, ma-empower din yung ibang mga women scientists who are here. Ang pharmacy, puro babae yan, no? Ayan, so, yung, sabi nga ng mga students ko before, students ko, sabi nila, Sir, bakit ganun? Wala na namang pogi dun sa first year ng pharmacy. Puro babae yung pumapasok, no? Tapos tapos ginagawa nila, sumisilip sila sa labas ng building, no? Kasi yung ibang colleges, no, for example, rehab science, no, radtech, maraming lalaki doon, no? Ayan, so sumisilip sila. Kasi wala raw lalaki sa pharmacy, no? Ayan. So kaya i-highlight natin si Rosalind Franklin. Okay? So nagets niyo? So kaya ko to sinabi dito, no? Ang dami ko na sinabi about secondary structure, sana nakuha nyo. Ayan, so number 1, the base pairing, no, complementary. Adenine forms complementary base pair with thymine. The hydrogen bonds, I forgot to mention dalawa, no? So that there are 2 hydrogen bonds between adenine and thymine. And as you can see in this diagram, ayan. Tapos for guanine and cytosine, there are 3 hydrogen bonds. Okay? Ayan na na-form. Another thing that I mentioned regarding secondary structure ay yung double helical structure. Okay? So, double helix siya. So, in fact, siya ay, based on its helical structure, it can, teka lang, lipat na tayo sa next slide. Ayan. It can actually form, no, a right-handed helix, no? Ay, magbura tayo. So, it can form right-handed helix. So, for example, yung ADNA and yung BDNA, both of these are right-handed helix. Okay? So, pag sinabing right-handed helix, clockwise yung rotation. Okay? Tapos, the ZDNA is the only left-handed helix. Also, remember that the backbone is sugar phosphate. Okay? Tapos, in the middle, you have the bases. They are stacked like staircase. No? Magkakapatong sila. Okay? The bases, no? The bases are perpendicular to the helical axis. Okay, yan. So tandaan, the bases are perpendicular to the helical axis. Okay, yan. So perpendicular, nakaganon. Okay? Yan. That's why certain anti-cancer drugs can intercalate in between the bases. Okay? Ayan. Another thing with regards to the secondary structure of the DNA is that if you place the DNA under physiologic conditions, what you will get is the B form of the DNA. Ito yung nakita ni Rosalind Franklin. Okay? Now, if you dehydrate, no, if you dehydrate or you place the DNA in alcoholic solutions, you will get an DNA. Ito siya, no? Ayan, mas punggok yung DNA. And then, certain areas of the genome, particularly yung tinatawag nating CPG islands, okay, would have the zDNA. Ito yung highly regulated na portion of the genomes. No? You will find the zDNA. It is the only left-handed structure or conformation of the DNA. Alright? Ayan. So, yan yung mga sa DNA. Now, sometimes, you don't have two strands that are separate, okay, forming a double helix. Okay? Sometimes it's one strand only, isang strand lang, tapos magpo-form ka ng double helix. Okay? Ayan. Or magpo-form ka ng base pairing, no, in one strand. This happens if you have a palindromic sequence. Ayan. Nakikita ba dyan? Hindi nakikita. Okay, so sige, sulat natin. So, palindromic sequence. Palindromic sequence. Ayan. So what is this palindromic sequence? The palindromic sequence is a sequence, no, na inverse complement of each other. Okay? In the same strand. They are inverse complement of each other on the same strand. So, anong ibig sabihin ng inverse complement on the same strand? So, para mas malinaw, no, mag-drawing na lang ako sa whiteboard. Hold on. Ayan. Okay, kita naman siya. Both. Okay, lalakihan ko yung sulat ko. Okay. So, what is palindrome? So, basically, ganito siya, no? You have one strand. Ayan, isang strand lang. Tapos, meron kang inverse complement on the same strand. Okay? So, for example, meron akong G, A, C, T, T, ano pa? A, sige, C, T, A. Ayan. Tapos, G, T, A, G, T, C. Ayan, okay? So let's suppose I have this sequence. Ayan. So kunwari may ganito akong sequence. Ang haba, no? Para masaya. GAC, TTA, CTA, GTA, AGTC. Alright? Now, if I examine this very closely, okay? Ito yung mapapansin ko. Yung T at saka yung A, these are complementary. Right? Okay, complementary yung A at saka yung T. Okay? Yung C at saka yung G ay complementary. Okay? Yung A at saka yung T ay complementary. Yung T at saka yung A, complementary. Same with this one, complementary din. Yung C at saka yung G dito, complementary din. Yung A at saka yung T, complementary. Yung G at saka yung C, complementary. Alright? That is a palindrome. Okay? Ayan. So, ibig sabihin, this strand can fold. No? Pwede siyang mag-fold. Ayan. This can fold like this. Okay? Mag-fold siya ng ganito kung saan andito yung T, andito yung C. Ayan, paatras ako. Ito. Ito. T, C, A, T, T, C, A, G. Okay? And then, sa kabilang side naman, ayan, pag ganito tayo, ayan. So, you have your A, G, T, A, A, G, T, C. Okay? Ayan. So, that is a And that is an example of, ayan, a palindrome. Ayan, so ito ay mag-complement. Okay? So, pwede siyang mag-fold on itself. No? The strand, no? Single strand can fold on itself because of the palindromic sequence. Okay? Ayan. So, sino dito yung nakabasa na ng ano yun? The Curious Case of a Dog in the Nighttime. It's a book. Ayan. So, ayan. So, so it's about an autistic boy, no? It's about an autistic boy, no? Si ano kasi yung character na yon, whenever he sees, no, cars na kulay red, binibilang niya, no? Ayan, kasi parang may relation daw sa kanyang life yung mga cars na red, no? And marami pa siyang ibang ginagawa, no? Like for example, he looks at the plate numbers, no, or he looks at numbers and then iniisip niya kung palindromic ba yung numbers. Okay? Ayan. So for ano ba yung palindromic na number? So for example, yung number ay 1, 0, 2, 3, 5, 3, 2, 0, 1. Ayan. So this is a palindromic number. Okay? Baliktaran, pareho siya. No? Ayan. Hatiin natin sa gitna. Yan. Ito yung gitna. Ito pareho. Ito pareho. Ito pareho. Ito pareho. No? Ayan. So sometimes, no, ganyan din ako, no? When I look at telephone numbers, birthdays, days of the week, no? Una ko iniisip, palindromic ba yung number? No? Ayan. So, bigla ko iniisip, no? Ayan. So, isip ka kung ng palindromic na date, no? Baka may importance 'yun sa iyo, no? Baka doon mo mag-meet yung crush mo or doon kayo magde-date, no? Hindi ko alam. Basta 'yan, no? Ayan. So, that's an example of a palindromic number. Pag number, madali lang, no? You just fold the number. Pag pareho sila, yun na yun, palindromic. Okay? Pero for palindromic sequences, no? Remember, inverse complements. No? Ayan, complement sila. Okay? They are not the same. Okay? Nagets? Ayan. So that is a palindromic sequence. All right. Now, balik lang tayo dun sa slides ulit. Ay. All right. So when you have the palindromic sequence will not only produce a secondary structure for a single strand. Pwede rin siya sa double strand. Okay? So pag may dalawang strands, ganun din. Pwede rin magkaroon ng palindromic sequence. Okay? So this palindromic sequences can form folds we call hairpin. Okay? Tulad ng pinakita ko kanina, hairpin yun. Hairpin means pag ganito, no? Yung mga hindi nakakakita, ayan. Pag ganito, hairpin. Okay, yan. So hairpin form. So dahil nagkaroon ng palindromic sequence, nag-fold siya on itself, forming a hairpin. Or pwede rin, dalawang hairpins. Ayan. This is what you call a cruciform. Okay, mukhang cruise. No, ayan. Formang cruise. No, ayan. Kaya cruciform. Alright? Ayan. So, yan yung effect ng palindromic sequences. All right. Okay. Yeah, so next is, let's proceed to the next part, which is all about the tertiary structure. Ayan. So what is the tertiary structure? The tertiary structure is the packing of helices, bulges, and single-stranded coils into a stable and more or less compact body. Okay? So this includes, no, yung tinatawag nating supercoiling. Ayan. So familiar kayo with the term supercoiling? Ayan. So para madaling maalala kung ano ang supercoiling, medyo mahirap i-supercoil to. Ayan. Yan, I know and I understand that a lot of you do not know what cord telephone looks like, no? Meron pa ba dito na abutan ninyo yung may cord na telepono? Meron pa? Sure. Hindi kayo nananaginip. Ayan. Okay. Ayan. Sa school. Ang saklap naman, no? Ibig sabihin, napakaluma ng school na yan. Okay. So yung may cordon kasi na telepono, di ba? One of the major problem there is yung pagbumuhol-buhol yung telepono, di ba? Ayan, kasi nga, supercoiled yung telepono, no? Yeah, now let me show you how supercoiling actually happens. Ayan, teka, hindi ko siya ma-uncoil. Ayan. So, teka lang. Try kong i-demonstrate sa harap ng camera. Okay. So, oops, sana wala pong sumabog na cord. Okay. So, this is your double helix. Ayan. Teka, hindi muna ako magmamic ha. May microphone naman for the online students. Ayan. Okay, so this is your double helical or your duplex DNA, for example, no? Yan. So, remember na helical na siya in structure. Okay? Yan. So, helical na siya in structure. So, ganito na yung itsura niya. Okay? Now, if this continues to fold, no? Ano yung susunod na mangyayari, no? Yeah, so ikot-ikuti mo pa siya ng paulit-ulit, no? Ayan, so it will naturally coil on itself, no? Ayan, so teka lang, ang hirap ng iikot. Matigas kasi. Ayan. So kung iikot-ikutin ko to, ayan, iikot siya on itself, no? Ayan, pipihit ko pa. Ayan. Ayan. Pihit pa natin. Ayan. So umiikot siya ng mag-isa. Okay? Ayan. So that is super coiling. Okay? Ayan. Umiikot siya. Now, what's the effect of super coiling? Ayan. From a very large molecule, okay, lumiliit siya. It becomes compact. Okay? Ayan. So super coiling is essential to condense, no? To condense or to make your genome compact. All right? Ayan. So now there's a problem. Okay? In a supercoiled DNA, for example, this one, okay, the transcription factors, no, cannot reach the genes. Okay, transcription factors are proteins. These are large proteins which bind to the DNA para ma-convert yung DNA to mRNA. Okay, so dahil naka-coil yung inyong DNA, hindi siya makadikit ngayon doon sa gene, nakatago siya. Okay, yeah, so nakatago, no? So dahil nakatago siya, it cannot be transcribed, no? In other words, it cannot be expressed, no? The gene cannot be expressed. The gene is turned off. Okay? Ngayon, yung mga genes na nandoon sa hindi coiled area of the DNA andito, okay? Madali siyang lapitan ng transcription factor. Okay? So these genes are turned on. Okay? Yeah, so these are turned on. Okay, so ibig sabihin they can be expressed, those genes. Alright, nagets? Ayan. So yan yung purpose ng supercoiling. Ayan. Now, you can actually relax the supercoiling by cutting, no? By cutting the supercoiled DNA. Alright? And remember that DNA is called a relaxed DNA, no? Ayan. Pag ito, ayan, binitawan ko 'yon, no? Mas maganda sana kung rubber band eh. Ayan, kung may rubber band lang ako, ayan. So pag binitawan ko 'to, bubukas siya mag-isa, no? Ayan, that is a relaxed DNA. All right? Ayan. So now, take note na these conformations of DNA are what you call your DNA topology. Okay? DNA topology. It is the degree of supercoiling of the DNA strand. Okay? Now, DNA molecules, no, which only vary in their topology are known as topoisomers. Okay? And you can interconvert between topoisomers through topoisomerases. All right? So anong ibig sabihin nun? Let's say you have a positively supercoiled DNA and you want to convert it into a negatively supercoiled DNA, you need a topoisomerase. I-unwind ng topoisomerase yung supercoiled DNA to form, no, the negatively supercoiled DNA. All right? Ayan. Now, these enzymes are known as DNA topoisomerases. Okay? The DNA topoisomerases are also known as, ayan, other name, DNA gyrases. Ayan. Ayan, DNA gyrases or gyrases. Alright? Take note that some drugs inhibit the DNA gyrases. Okay? So pag in-inhibit yung DNA gyrase, ano yung mangyayari? Dahil na-inhibit yung DNA gyrase, hindi ma-uncoil yung supercoiled DNA. No? Pag hindi na uncoiled, yung supercoiled DNA, anong effect? Yung gene ay hindi matatranscribe, hindi siya may express. The cell reproduction stops. No? Kaya siya nagiging anti-cancer agent. Nagets? Ayan, yun. No? Ayan, kaya may kinalaman siya dun sa DNA supercoiling. Alright? Ayan. Now, there are two types of topoisomerases. You have your type 1 topoisomerases and type 2 topoisomerases. So, type 1 cuts one strand. Type 2 cuts two strands. Okay? And the following are the anti-cancer agents which are known as your topoisomerase inhibitors. Ayan. As ito, favorite ko rin to. Yung quaternary structure. Okay? Now, what is this quaternary structure? So, remember, may binanggit na ako kanina, no? That histone protein, no? The histone protein binds with the DNA to form what you call the nucleosome. Ayan. This is the nucleosome. Teka lang, ha? Ayaw niyo lumaki. Okay. So, this is your nucleosome. The nucleosome is composed of 8 histone molecules and DNA wrapped around the nucleosome, no? Mukha siyang mukha siyang beads, no? Ayan. So, yung histone proteins, no? The histone proteins are known to have a lot of these amino acid. Ay, teka lang. Ay. Ayan. Your histone protein has a lot of lysine. Okay? Now, lysine, no, i-discuss pa natin siya later or tomorrow na siguro. Lysine is a positively charged amino acid. Ayan. It is a positively charged amino acid. Okay? Now, yung lysine ay positively charged. Okay? So, kung marami yung histone ng lysine, what is the charge of histone protein? Positive, no? Ayan, positively charged din siya. Yung DNA kaya, anong charge? Alright? Nice question. Anong charge ng DNA? Ulitin natin, ha? Saan gawa ang DNA? May base, may sugar, may phosphate group. No? Ayan, may phosphate group. You have the sugar phosphate at the backbone. Anong charge ng DNA? Positive o negative? Diba? Ang dulo natin, ang suffix natin ay 8, no? Ayan, 8. Parang acetic acid, acetate. Diba? Acetic acid is the acid form, acetate is the base form. Ibig sabihin, negatively charged. Okay? So, the DNA is negatively charged. Okay? So, positive yung histone, negative yung DNA, the ionic interactions between the DNA and the lysine molecules in the histone proteins are the ones that compact your nucleosome. Siya yung dahilan kung ba't umiikot yung DNA sa histone proteins. Okay? Ayan. And then you can just imagine na marami yun, no? Umiikot-ikot yung DNA mo doon sa nucleosome. Okay? Now, just like supercoiling, may problema tayo dito. Okay? May problema tayo dyan. Okay? Ayan. Because the DNA is wound around the histone proteins, they are not available for transcription. No? Hindi sila available for transcription and even replication. Hindi. Nakadikit kasi sila. No? Unavailable na sila. Alright? Ayan. So these are what you call the heterochromatin. Heterochromatin. Heterochromatin, ito yung densely packed portion of your chromatin. No? Usually, ito yung mga nakapalibot doon sa histone proteins. Okay? These are genes that are switched off. Okay? Heterochromatin. Now, yung DNA naman na hindi nakapaikot, for example ito, no, na hindi nakapaikot doon sa histone proteins, yun ay tinatawag natin na euchromatin. Euchromatin. Okay? Yung euchromatin, ito yung available na genes. They are switched on. Okay? Yan. So, kung ito ay off, kung yung heterochromatin ay off, ayan, yung ating euchromatin ay on. Alright? Ano pa yung pwedeng sabihin dito? Okay, now, there are certain modifications to the histone, no? Proteins. Meron tayong tinatawag na histone acetyltransferase or HAT. Okay? Histone acetyltransferase or HAT. Okay? The histone acetyltransferase would transfer an acetyl group to the lysine. Okay? So yung lysine, kakabitan mo ng acetyl group. Now, this will reduce the interaction between DNA and the histone protein. Kasi nawala yung ionic interaction. Okay? Ayan. So, kakawala yung DNA from the histone if you have a histone acetyl transferase. Alright? Ayan. So, ibig sabihin, yung gene na yun na kumawala sa histone will become euchromatin. Okay? It will be turned on. Okay? Ayan. And then, meron din tayong tinatawag na histone deacetylase enzyme. Deacetylase enzyme. Kabaliktaran naman to. Kung yung histone acetyltransferase transfers acetyl group to the lysine, the histone deacetylase removes the acetyl group from the acetylated lysine. So anong effect nito? Kabaliktaran din, no? Magiging positive yung lysine, so the DNA will again go back, no? Makikipag-interact kay histone. Magiging turned off yung gene. Gets? Okay? So si HAT will turn on your gene, si histone deacetylase will turn off your gene. Alright? Ayan, so yan yung tinatawag natin na epigenetic modifications. Yan, dito ko na lang sulat. Epigenetic modifications. Alright? Yan, epigenetics. Alright? Ayan. So actually, it's now the more modern approach to cancer therapy. No? Ayan, epigenetics. Ayan, or even other mga genetic diseases. Alright? You don't have to knock off or remove a gene. You just have to control how the gene is expressed. That is epigenetics. No? Controlling how a gene is expressed. Epigenetics. Alright. So now we proceed to, ayan. Ay, ang daming nakasulat. Okay, so magbura tayo. Okay, so properties of your nucleic acids. Ayan. So basically, you have denaturation of the DNA and even RNA duplex. All right. Now, remember when you say denaturation, okay, it's just the breaking of non-covalent bonds. Okay, yeah. So tandaan niyo palagi pag sinabing denaturation, whether it is protein, whether it is nucleic acid, it is just the breaking of non-covalent bonds or yung tinatawag nating intermolecular forces of attraction. Okay? For nucleic acids, denaturation involves the removal, or the breaking of the hydrogen bonds, H bonds. Okay? Ayan. So, maghihiwalay ngayon yung complementary bases. No? That is denaturation. Now, remember then that nucleic acids are more stable than proteins. No? Nucleic acids can withstand temperatures up to around 100 degrees Celsius. Okay? Hindi sila agad-agad masisira or magde-denature. No? Unlike the proteins, at around 60 degrees Celsius, some proteins already coagulate, they already denature. Alright? Ayan. So kung mag-isip kayo between the properties of the two biomolecules. Now, aside from temperature, very low salt concentration, or even extreme pH, like acidity or basicity, can also affect the breaking of the complementary base pairs in your DNA, particularly. Now, we have this term which is known as melting temperature. Okay, melting temperature. Ayan. Familiar, no? Familiar naman sa melting temperature. Ayan. So, sige, how do you define? Sabi nila, pag dinefine mo raw yung melting temperature, malalaman mo daw no, kung anong level ng maturity ng isang tao, no? Ayan. So, sige, magtatanong tayo. Kasi bila sila natakot, no? Nakita ko yung mga takot sa kanilang mga mata, no? Ayan. Nahiya silang sabihin, no? Nahiya silang i-reveal yung maturity level nila, okay? Ayan. So, let me explain why. When I ask, no, what is melting temperature, ang kadalasang sagot, no, ng high school or ng elementary ay ganito. It is the temperature at which a solid becomes liquid. Tama naman. Tama naman, di ba? Ayan, so it's the temperature at which solid becomes liquid. Melting temperature, okay? Kaso, when you are already in college, no, iba na, no? Iba na yung definition na binibigay sa atin, no? Sometimes, no, kasalanan din ng mga college teachers, no? Bakit kasi yung mga college teachers, tulad ko, no, ang dami kong dinadaldal, ang komplikado ng mga terminologies, ba't hindi na lang tayo mag-stick dun sa mas madaling definition? Okay? So anyways, melting temperature, as another definition, no, it is the temperature at which the crystal lattice of the substance, no, hindi, ulitin natin. Ulitin. Ayan. Melting temperature is the temperature at which the energy is sufficient to break the crystal lattice structure of the substance. Okay, ulitin natin yon. Melting temperature is the temperature at which, no, or it is the temperature at which the energy, no, of the system is sufficient to break the crystal lattice structure of the molecule. In other words, the solid becomes liquid. Okay? Ayun. So, di ba yun lang naman yun? Pag nasira yung crystal lattice structure ng substance, ang ibig sabihin lang nun, no, naging liquid yung solid. Yun na yun. Di ba? Pinahirapan mo lang yung sarili mo. Ayan. So, that's the definition. However, for nucleic acids, the term melting temperature is quite different. No? Ayan. It's different. Melting temperature is the temperature at which 50% of the DNA strand, no, is single-stranded. Ulitin natin, no? Yeah, so for nucleic acids, no, remember, melting temperature is the temperature at which 50% of the DNA molecule is single-stranded. Okay? Ayan. Now you may ask, sir, bakit hindi na lang yung, hindi na lang completely single-stranded. Okay? Diba? Ba't hindi, no? Weird din talaga sila mag-define, no, ng mga melting temperature. It's because, no, it's quite difficult to know, no? It's quite difficult to know whether a DNA molecule is already fully single-stranded. Okay? It's much easier to determine at 50%, no, of the DNA molecule. All right? Ayan. Now, ba't ko sinabi yun? Kasi, the melting temperature is related to denaturation. The reason why single strands are formed from double-stranded DNA ay dahil sa breaking of the hydrogen bonds. And that is denaturation. Okay? Ayan. Now, take note that phosphodiester bonds between sugar phosphates are not denatured or are not cleaved. They are not broken kapag denaturation. Okay? Ulitin natin yon. Phosphodiester bonds are not broken during denaturation. Okay? Only hydrogen bonds. Okay? Now, what do we again know about the complementary base pairs of your bases? Complementary pairs of your bases. We know na 80, no, adenine and thymine. Teka lang. Oops, lumabas ka. Okay. Remember that adenine and thymine form two hydrogen bonds. Okay? Yan, sabi ni Chargaff to. No? And then guanine and cytosine form 3 hydrogen bonds. Okay? So, between the two, sino yung stronger pair? Si AT or si GC? Ang stronger pair ay si GC, correct, no? So, this is stronger. Okay? So, if a molecule has higher GC content, no? Pag mataas yung GC content, the melting temperature, no? The melting temperature of the DNA is higher. Okay? Ayan. So again, if the DNA molecule has a high GC content, then the melting temperature is higher, no, than one with high adenine thymine content. Alright? Ayan. So yan lang naman yung patungkol dito. Okay? Ayan. So GC content is an important factor to determine the melting temperature of your DNA. Ayan. So may 30 minutes pa. So puntahan na natin yung central dogma. Okay? Ayan. Yung madugupang topic, no, for the last 30 minutes. Okay. So ayan. So the central dogma. So pag wala kasing synthesis ang nucleic acids, no? Maliban dito. Ayan. All right. So central dogma states that the DNA is the gene. No? It's the primary gene. Although even the RNA can be considered as a gene. Okay? Ayan. But most of the biomolecules in the body, no, are synthesized according to the instructions from the DNA. No? Ayan. Kaya siya yung tinatawag natin talaga na gene or source of instruction. Okay? Now, the DNA is replicated, no? Ayan, or pinaparami, no? The number of DNA molecules multiply in a process called replication. Okay? The DNA is then transcribed or converted to mRNA, messenger RNA, no? Through process called transcription. Okay? And then the mRNA is then converted into a protein through translation. Okay? So that's the central dogma, no? So you have DNA replication, transcription, and then finally your translation. Remember that gene is the basic hereditary unit that contains the information for making the RNA or polypeptide. Now take note that in this definition, sinama ko yung for making an RNA, no? Because the gene is also responsible for making your tRNA, your ribosomal RNA, rRNA, no? Aside from the messenger RNA, okay? Yan. Yung tRNA at saka yung ribosomal RNA, they don't become proteins, no? Hanggang doon lang sila, okay? Only the mRNA is translated into a polypeptide or a protein, okay? Ayan. Yeah, so we already mentioned this, no, in before, no, the difference between prokaryotic and eukaryotic na replication, no. So basically yun lang yun. Ang hindi pa natin na-discuss dito ay yung introns, okay? Now remember na in prokaryotes, there is simultaneous transcription and translation, okay? So the mRNA transcript, no, the mRNA transcript of prokaryotes ay mature. Okay? It doesn't need splicing, no? Hindi na siya kailangan cut pa. Okay? However, for eukaryotes, it's different. The mRNA will be transported from the nucleus into the cytosol, okay, or the cytoplasm. In the cytoplasm, there are a lot of things that can destroy the mRNA, okay? And because of that, nilalagyan muna ng mga modifications yung mRNA, okay? And we call that the pre-mRNA, okay, to withstand, no, the cytosolic environment, okay? Aside from that, eukaryotic transcripts would contain introns, okay? What are these introns? The introns are intervening sequences. Ayan, intervening sequences. Ayan, so these intervening sequences are removed, no? Ayan, they are removed through a process we call splicing na gumagamit ng spliceosome. Ayan. So basically, ang itsura nun is here you have a an mRNA transcript. Okay? So this ones, no, ayan, ito. Kung nakikita niyo yung drawing ko, ayan, na cute. Ayan. Yung box, yan yung mga intervening sequences. Hindi yan kailangan in the mature mRNA, no? So what is done, no, doon sa introns na yan, they are removed, no? They are eliminated. Ayan, so they are removed. Ayan, tatanggalin sila ng spliceosomes. Ayan. Tapos, what happens to them is, ayan, pagdidikitin ulit itong mga ito, forming your mature mRNA. Okay? Now, intron removal or processing of the pre-mRNA happens only in eukaryotic organisms. Okay? Ayan. So, basically, yan lang yung gusto kong i-highlight in this particular slide. Okay. Now, in this slide, what I want to highlight is this, no? A lot of targets, no? A lot of targets of drugs, particularly for cancer or against cancer and against viruses ay nag-a-attack ng central dogma, no? Pwedeng i-attack yung DNA replication. Okay, so for example, these ones, to attack the DNA replication, you use an analog of the base, no? Akala ng viral or cancer cell, okay? Akala ng viral particle or cancer cell, nag-incorporate sila ng tamang base, no? Ayan. Pero mali palang base yung ini-incorporate nila. Okay? Ayan. So dahil mali yung base na nilagay nila, they would fail in replication. Hihinto, no? Yeah, so that's how you stop cancer or viral replication. Okay? Yun yung analogs of your bases, no? Your purines and your pyrimidines. Another way, no, is to inhibit the DNA gyrase, no? Ayan. So naalala niyo yung gyrase? It is your topoisomerase. So when you prevent the unwinding or the uncoiling of the DNA, you also prevent replication. Okay? Next, we have DNA-dependent RNA polymerase inhibitors. We also have yung inhibitors of your 50S or 30S ribosomal subunit in the prokaryotes, in bacteria. Okay? Now, meron din tayong tinatawag na reverse transcription. Okay? So what is this reverse transcription? Remember that in the central dogma, the DNA is transcribed to form mRNA, okay, messenger RNA, okay, which then is encoded or is translated into a protein or polypeptide. Okay. Now, in some viruses, no, they do not have DNA. They only have RNA, no, RNA lang. Ayan. So they would depend on the RNA, no, to produce a DNA molecule. Okay. Gumagamit sila ng tinatawag natin na RNA dependent. Ayan, RNA dependent. Ayan, DNA polymerase. Ayan. Ang other name nitong napakahabang pangalan na ito ay reverse transcriptase. Ito na yun. Okay? Ayan, so RNA-dependent DNA polymerase is simply your reverse transcriptase. Okay? Ayan. So, some viral particles kasi like retroviruses, no? Yung human immunodeficiency virus, HIV, wala siyang DNA, so yung RNA niya, i-convert niya muna into DNA. Then its DNA will then be incorporated in the DNA of the host. Okay? The host will replicate, no? Yung DNA, akala niya walang mali doon sa kanyang DNA, yung pala, meron nang in-insert yung virus na DNA, no? Doon sa kanyang DNA. And then, pagka-replicate, no? Yung DNA-dependent RNA polymerase naman will then convert the DNA from the host, no, na mag-encode siya ng viral RNA, okay, which will eventually produce the viral particles. Alright? Yan. So, kailangan natin i-inhibit din yung reverse transcription, no? Yan, by inhibiting the reverse transcriptase. Yeah, so those are the targets, no? Ayan, of a lot of drugs, no? Particularly antivirals and anti-cancer agents. Ayan. Actually, feeling ko matatapos naman tayo until replication. Okay? Bago mag-end yung 5 p.m. Okay, so let's define replication. So replication, no, basically it is the synthesis of new strands of DNA. Okay, remember that in DNA replication, the direction is 5 prime to 3 prime. Okay? Anong ibig sabihin ito, no? Ng 5 prime to 3 prime direction ng DNA replication. Let's suppose this is your 3 prime, 5 prime strand. Okay? And then, meron din akong, nakikita ba dyan? Nakikita nyo? Ayan. So, meron tayo dito na 5 prime, 3 prime strand. Okay? One strand will have a continuous replication. Okay? The other one will not have a continuous replication. Okay? Ayan. So, remember na 5 prime to 3 prime kasi yung direction natin. So, this one becomes your template strand. Yung 3 prime to 5 prime, that is your template strand. Okay? Yan, kasi yung movement natin ng polymerase is from 5 prime to 3 prime. Okay? And then the other strand, no, discontinuous yung kanyang replication. Okay? Ayan. Kasi pabaliktad, no, yung position niya, no? So we call it your non-template strand. Okay? Yan. So nagets yun? Important dito is that the template strand is the 3 prime to 5 prime. And then, nagpo-produce siya ng 5 prime to 3 prime direction ng DNA strand. Now, the replication is semi-conservative. No? Ayan. So what do we mean by semi-conservative? The products of replication would always be a hybrid. No? One strand comes from the parent strand, the other strand is a daughter strand. Okay? So yung isa ay parent, yung isa ay daughter palagi. For example, yung dinowing ko pa lang, and over here, ayan. Okay? So this is one pair. Okay? Yan yung isang mapoproduce natin na double strand. Okay? One is original, yung kulay red. Okay? That's the parent strand. And then the other strand is new. Okay? So that's the daughter strand. So it's semi-conservative. Alright? And then lastly, it's discontinuous on the non-template strand. Okay? Because of the position of the DNA polymerase. Alright? Ayan. So, simulan natin doon sa pre-priming complex. And what is this pre-priming complex? What has been elucidated very well ay yung prokaryotic replication. No? Hindi to eukaryotic. No? Medyo complicated yung eukaryotic replication. But we assume that similar lang yung eukaryotic and prokaryotic replication. Alright? Now, the pre-priming complex contains the DNA protein, which melts the AT-rich region, the DNA helicase, which unwinds, no, the double-stranded helix, and the single-stranded DNA binding proteins, which keep the double-stranded DNA separate. Okay? So basically, no, para siyang zipper, no? So your DNA helicase, no, it's like the yung thing na hiyagano mo sa zipper. Okay? Ikaw alam ko ang tawag doon. Yeah, so yung sa zipper, di ba, may pang-unzip ka. Okay? That's your DNA helicase. Okay? Ina-unzip niya, no, yung double helix. Alright? Yan. Tapos, the other one, your single-stranded binding protein will keep the two strands separated. Okay? Kasi, pag wala yun, pagdidikit ulit sila, no? Spontaneously. Okay? Reverse of denaturation. Now, remember that in DNA synthesis, the first thing that happens is the incorporation of an RNA primer. Okay? The RNA primer basically signals where to begin the replication. Okay? Now, the RNA primer binds on the template strand, the 3' to 5' strand. So, for example, this is the template strand. Ayan. Ay, kulay yellow yung ginamit ko, so hindi nyo siya nakita. Ayan. So, this is the template strand. Ayan. Dito kumabit yung inyong RNA primer. Nandito siya. Okay? Ayan. Now, what happens is, as you go along, no? As you go along, or as the DNA polymerase moves along the strand, no, of the DNA, nagkakabit siya ng nucleotides in the template strand, no, complementary to the bases of your template strand. Okay? There's a problem. Okay? There's a problem. Dito, dire-diretso siya. No? Ayan, dire-diretso siya doon sa template strand. We also call that the leading strand kasi dire-diretso siya. No? It's continuous. Okay? Sa kabilang side, may problema siya. No? As you can see here, baliktad. No? Pag 5' to 3', gaganon ako dapat. Pag ganun. Pag ganun. No? Ayan. So I can never have a continuous synthesis on the other strand. Okay? Yan. So kailangan, no? Ang gagawin ko is I will again put your RNA primer. So kunwari somewhere dito may RNA primer. Ayan. And then magdadagdag siya ng nucleotides pabaliktad. Okay? On that same position. So you can just imagine, no, na pag ganito siya. Yeah. Ayan. So ganito, no? So let's suppose ito yung template strand, okay? Tapos ito yung non-template strand, okay? Tapos this is your pre-priming complex, no? Containing your DNA helicase. Pupunta siya dito, ibubukaka niya yan, no? Ayan. Ayan. Tapos it will keep these two strands separated, okay? So this is your template strand. Your DNA polymerase is over here. Ang ginagawa ng DNA polymerase is nag-add siya dito ng bases, okay? Dire-diretso siya. Pero pagdating dito sa kabila, no, since dapat 5' to 3' yung direction, ang gagawin niya is pag ganito siya magdagdag. Okay? So dito, from the RNA primer, pabaliktad siya pag ganon. Okay? Tapos dito, pag ganon siya. So as it moves along, okay, it will leave fragments, okay, in the non-template strand, okay, also known as the lagging strand. Okay? This is the leading strand, this is the lagging strand. So habang gumagalaw yung DNA polymerase, no, ito namang pre-priming complex umaatras. Kasi ito yung DNA polymerase. So umaatras ito nagdadagdag dito. Alright? And imagine, that's how replication happens. Ayan. So ang dapat tandaan dito, no, is that, again, meron tayong pre-priming complex, meron tayong RNA primer, okay? At the RNA primer is where new nucleotides are added, no? Ayan, okay? Gets? Tapos you have the leading strand, which is continuous, okay? And then you have the lagging strand, which gives you the Okazaki fragments, okay? Putol-putol yung synthesis in the lagging strand. All right? Ayan. Now, take note that there are two types of polymerases in prokaryotic replication. You have the DNA polymerase 3 and DNA polymerase 1. Okay? The main polymerase that adds nucleotides, no, complementary to the template strand is your DNA polymerase 3. Okay? The DNA polymerase 3 has the 5' to 3' polymerase activity, which adds the bases from the RNA primer. Okay? On both the leading strand and the lagging strand. Okay, so take note of that. Maraming nalilito doon. Ayan, so it adds bases on both the leading strand and the lagging strand. Okay, now, remember na meron tayong Okazaki fragments. Okay, so may Okazaki fragments tayo, and in order for you, no, to fully synthesize the other strand in the lagging strand, okay, you have to remove the RNA primer. For the leading strand, wala tayong masyadong problema, no? Kasi sa leading strand, we only have one RNA primer, di ba? Kasi isang beses lang niyang ikakabit yung primer. Doon sa lagging strand, every time nagagalaw yung DNA polymerase, maglalagay siya ng RNA primer. Okay? Ayan. So maraming RNA primers in the lagging strand. Okay? So now, in order to have a complete duplex DNA, you have to remove the RNA primer, okay? And this is through the DNA polymerase 1, okay? Take note that DNA polymerase 1 has the 5' to 3' exonuclease activity, no? Ayan. So meron siyang 5' to 3' exonuclease activity, which is important in its removal of the RNA primers. Okay? Now, both DNA polymerase 3 and DNA polymerase 1 has the 3' to 5' exonuclease activity. So, ano naman yung ibig sabihin nito? The 3' to 5' exonuclease activity are important for proofreading. Ayan, proofreading. In other words, di ba gumagalaw siya 5' to 3'. Okay? Na-realize niya, ay teka lang, mali yung base doon sa likod. Aatras siya. Okay? So atras siya, tatanggalin niya yung base, no, na nailagay niya. No? Parang ikaw, pag nagsusulat, ay mali yung spelling ko, bura. Okay? Ayan. So that's your 3' to 5', kaya paatras. Okay? Kasi aatras siya, tatanggalin niya yung wrong base. Okay? Then papalitan niya ng tamang base. Okay? This proofreading ability is very important, no, to maintain the sequence of a DNA. Okay? Kapag walang proofreading activity, ano yung magiging effect nito? There will be mutations. Okay? So this proofreading activity prevents mutations. Okay? Ayan. So both DNA polymerase 3 and DNA polymerase 1 has the 3' to 5' exonuclease activity. Ayan. Next, ayan. So ganito na yung mangyayari, no? Explain ko na yung mangyayari sa lagging strand. So natapos na ikabit yung Okazaki fragments. What will happen next? Okay? Number 1, RNA primers will be removed by DNA polymerase 1. Okay? Using its 5' to 3' exonuclease property or activity. Okay? And then after that, no, it will also fill the gaps. No? It will also fill the gaps na pinalitan ng RNA primers. So, for example, these are the RNA primers. Kasi ito, yung mga kulay blue. Ayan. These are the RNA primers. Papalitan siya ng tama na bases. Remember, magkaiba ang bases ng DNA sa RNA. Okay? Ayan. So, the gaps will be filled by DNA polymerase 1 again. Okay? And then, once finil niya yon, hindi pa tayo tapos, no? Kasi in the Okazaki fragments, putol-putol niya yung fragments. Ganito siya, no? Sa Okazaki fragments, ito yung RNA primers. So, ayan, RNA primers. Tapos, ito yung newly synthesized DNA. Ayan. Newly synthesized DNA. Ayan. Okay? Tatanggalin natin yung, ayan, yung RNAs. Ayan. Papalitan natin siya ng DNA. Ayan. Ibahin natin yung kulay. Ayan. Wait lang, malapit na. Ayan. Okay. Tapos, ayan. So, ito na yung DNA. Okay? Tapos, take note na at this point over here, ayan, iba namang kulay. Ayan. Putol-putol pa rin yan. Okay? Ayan. So, those nicks, no, those nicks will then be combined by DNA ligase. Okay? Ayan. So, the DNA ligase will connect the nicks formed, no, in the Okazaki fragments, no? Completing now your DNA. So ito mako-connect na natin siya. All right? Ayan. So teka lang. Exonucleases, anong ibig sabihin nun? Sa labas nagki-cleave. Okay? So for example, no, this is the DNA, dito siya mag-start mag-cleave or dito siya mag-start mag-cleave. Okay? So if this is 5' and this is 3', this is your 3' to 5' exonuclease property. Pag dito, that's your 5' to 3' exonuclease property. Alright? Now, kapag sinabing endonuclease, it's different. Okay? Pag endonuclease, sa loob natin siya i-cleave. So, for example, here and here, no? I-cleave siya. Okay? And ang tawag natin dyan, commonly ay restriction endonucleases, no? Especially if they have a recognition site, no? Other than the cleavage site, no? So we call them restriction endonucleases. They are important no, in the, cleaving or cutting DNAs, no? Ayan, DNA molecules. Ayan, last 2 minutes. Ayan. Ayan. So we have the eukaryotic polymerases. Okay? Now, eukaryotic polymerases, no, are different from prokaryotic polymerases. Sa prokaryotic polymerase kasi, dalawa lang yun. DNA polymerase 1 and DNA polymerase 3. Okay? Remember that DNA polymerase 3 is the main polymerase, no, in the prokaryotes that adds the bases. DNA polymerase 1 only replaces the RNA primer, no, in Okazaki fragments, no, and also in the leading strand, okay? And it also removes the RNA primer, okay, because of its 3' to 5' exonuclease, 5' to 3' exonuclease property. All right? Ayan. For eukaryotes, iba. Ito siya. Okay? Marami yan. Polymerase alpha, beta, gamma, delta, and epsilon. Pero don't worry, hindi to lumalabas, no? Kasi these functions, no, are not yet really fully resolved, no? Hindi pa natin sure kung totoo yung mga yan, okay? Kasi it's difficult to study eukaryotic replication, no, compared to prokaryotic replication, all right? But you will see na andito rin yan lahat. There's the primase, no, which adds the RNA primer. There's gap filling, which is done by DNA polymerase 1, okay? There is leading strand and lagging strand polymerase, no, done by polymerase 3. Okay, ayan. So nandiyan pa rin naman sila. Okay, may isa pang term which you might actually encounter, we call fidelity. Ayan. In DNA replication, the polymerase's proofreading property or yung kanyang ability, no, to religiously replicate the bases, no, ay very important property, yung proofreading property of the DNA to prevent mutation. There's another, no? Another important property of the polymerase is the fidelity. Anong ibig sabihin ng fidelity? Di ba sa tao, pag sinabing fidelity, laging magkasama. Okay? So, pag sinabi nating fidelity, okay, the DNA polymerase remains in the template strand, no? Hindi siya agad-agad umaalis, no? It does not easily, no, go out of the template strand, no? Yun yung fidelity. Okay? It remains attached to the template strand during DNA replication. Yes. Okay. I think that's the sign. Thank you, Siri. Ayan. So I think that's the sign, no, that we will have to end the day. Okay. Ayan. Okay. So ayan. So any questions so far? Ayan. Okay. So tomorrow, no? Tomorrow konti na lang din naman yung topic natin, no? Siguro the morning session will continue this one, plus amino acids, proteins, and peptides, plus the structure of carbohydrates, okay? Tapos in the afternoon, we'll have metabolism, okay? Ayan, so yun yung ating plan, okay? Hopefully, there will be no more problems tomorrow. All right, so if you have any other questions which you failed to ask today, no, okay lang, you may ask them tomorrow. All right? Ayan. Thank you so much, everyone. See you again tomorrow. Bye. Thanks. May nag-powder. Ayun na po. Ay, transfer. Ayan po. Stop share. Ikaw pa rin yung host, sir.